that is asexual reproduction in Spirogyra. Now, let's talk about the sexual reproduction. All right. Now, listen very carefully. When we took care of the paramecium sexual reproduction, we said two cells come together at the oral groove section to exchange chromosomal materials and sexual reproduction. In the case of Spirogyra sexual reproduction, that is also referred to as conjugation. So we must have two different filaments. So in the case of binary fission, we have a filament and a cell in the filament they divide, or we can have a lot of cells undergoing binary fission. Let's try this. So let's say binary fission, right? We got a long filament, let's say made up of 6 cells. Now, let me number them this time outside. Now, I'm going to ask a question about binary fission. Now, assuming for this 6-cell filament, binary fission occurred in cell 2 and cell 5. How many cells will result in the filament after the binary fission, Christian? Because I want to link it up to the conjugation.
Repeat the question again.
All right. Now, we had an original filament of 6. Cell 2 did undergo binary fission. Cell 5 also did undergo binary fission. How many cells will be present now in the filament after 2 and 5 cells did undergo the binary fission? I'm listening.
8 cells.
Okay, how did you get your 8?
The cell 2 splits to become 2 cells, and the 5 splits to become 2 cells. So if you count to be 8.
Okay, how many cells were added after the binary fission?
2.
2. Very good. So from what Christian is trying to say, so now we have cell 1, cell 2, cell 3, 4, 5, 6, 7, 8. So you can see that this computation is different from the one we did for fragmentation. So fragmentation can increase the filament numbers, but the cells are the same in terms of numbers. But in binary fission, we are having more cells being formed in one filament. So it was originally 6, now it is 8. And I'm very happy. Now let's go to the conjugation. So we have this time filament A and filament B. Let me pick a different color that is more conspicuous. So let's say this is filament A and this is filament B. Now watch the animation very carefully. We are having some additional information. Now you can observe that we have a minus and a plus. So you can see here we have a minus and we have a plus. And below we got a plus on the left this time and a minus on the right. Now when you check it very well, filament A, currently as it is being shown on the screen, got cell 1, 2, 3, 4. Then we got filament B, also having 1, 2, 3, 4. It can be more, but in this situation it's 4 each. Now when you check filament A, the materials in there are moving towards a cell in filament B. So let's see, let me pick another pen. Then let's see, I prefer different color. Pick white. So cell 1, 2, 3, 4. Now, check whatever is on the screen very carefully. I want you to understand the animation before we can go to the text. Once you get animation, everything is okay. Now, when you check cell 2 of both A and B. The cell content of cell 2 of A is moving towards the cell content of cell 2 of B. So because materials are moving from A to B, that particular cell giving out a cell content is referred to as the minus. And the one receiving it is termed as the plus or positive. When it comes to cell 3, this time we're having B releasing its cell content into A. So 3 of B is minus and 3 of A is plus. So in cell 4, this time we are having materials moving from A to B. So it means A4 is the minus and B4 will be doing the receiving, and that is referred to as the plus. So in real life, that is what actually occurs. Any cell losing its content to the adjacent filament cell is referred to as the minus, and the adjacent cell of the second filament receiving the material of the other filament is referred to as the plus. So let's go to the text. Now for conjugation, we must have two filaments lying side by side, or two conjugates. Secondly, we must have a protrusion forming towards each other. Watch it, I've stopped it. So when you check cell 2, we are having a protrusion from A getting towards B, and we are getting a protrusion from B towards A. Same for 3 out here, the protrusion from A towards B and B towards A. And for cell 4, same. So these protrusions grow towards the opposite cells. It means cell 2 will grow to cell 2. Cell 3 will grow towards cell 3 and not any other cell because they are very adjacent to each other. Now, when the two join, I repeat, when the two cells join, it is going to form a conjugation tube. When the two protrusions join, it is supposed to form what is called a conjugation tube. So now we have the conjugation tube being formed. Now, the cell content of the minus will shrink away from the cell wall to form a mass called the gamete. So after stopping this, check cell 2 of A, or filament A. Its cell content is moving away from the cell wall and is supposed to form a gamete. Same for 3 out here, and same for cell 4 out here as well. So once we are having the tube being formed and the mass has been moving or separated or shrunk away from the cell wall, and once we are having the tube called conjugation tube linking the two adjacent cells together, the materials in the minus will then move into the plus. So as the protrusion meets, the cell wall separating the two will split open because initially there was a cell wall. So let me stop it out here. So you can see that there was a wall out here, there was another wall out here. And once they meet, that wall must dissolve so that a tube will be formed with a space linking the two cells together. So that is what is occurring here and has just happened. And you can see that the materials in the minus is moving through the tube into the plus cell. So the cell wall breaks down, forming the tube linking the two cells. Now, this is conjugation occurring in real life. So this is an actual video footage of a Spirogyra cell having its content moving through the tube into the adjacent cell. So this is what actually occurs in real life. So gametes from one cell move to the other cell, and this time forming a zygote. In the case of the paramecium, we mentioned zygotic nucleus because it was the nucleus that did move. Over here, we have a stationary cell that ain't moving, but the content of the cell did move and fuse to the adjacent cell through the conjugation tube, and that will result in a formation of a zygote. Now, after this zygote has been formed, the plus will have a very thick resistant wall surrounding it. And that will lead to the formation of a zygote spore called zygospore. So when we have the mass of the minus moving into the plus, it is going to form a zygospore. And the zygospore is as a result of a thick wall around the plus cell, which did receive the content. And the reason conjugation occurs when the environmental conditions are not conducive. So, for instance, the water is drying up. So, conjugation must occur. And once we have the mass here and a thick outer protective covering is formed, it means that particular cell with that thick wall can stay on the dry aquatic environment until we get new water, so that it this time can germinate. The minus, which did lose its cell content, is no more existing, so basically it's dead, right? So the adjacent cell on the right, as you can see in the video, is also receiving content. So once the minuses release their materials into the plus, all the plus will have the wall formed around them, and any tube linking the two will disintegrate. Now, the zygospore that has been formed will germinate only under favorable environmental conditions, as the cell wall will split open and we are going to get cells coming out of it. And by cells, we are talking about a group of cells that are coming out to form a Spirogyra filament. Hence, cells emerge. I'm going to see some pictures of it soonest. But let's initially discuss significance. Before that, any question on conjugation?
Okay.
All right, so let's continue. Importance of the conjugation. Gametes are not exposed to the environment as they are well protected, because over here we did indicate that we'll get a thick resistant wall as the cell wall disintegrates. So the cell wall will be no more, but the thick resistance wall will protect it, and the zygospore is now well protected. It's called a zygospore, I repeat, because it is a zygote as a result of two cells fusing, that's the cell mass fusing, then it's just like a spore. That is how come it's called zygospore. So the zygote is protected. The gametes, that is the materials that move from the minus to the plus, are also protected. Then it brings about variation. Now, in biology, variation simply means differences among organisms of the same species. And you can remember when we took care of the paramecium, I said if we got a paramecium cell always undergoing binary fission, it means the chromosomes in paramecium A has now doubled up, resulting in two daughter paramecia cells. So it's always coming from a parent. But if we got two different parents exchanging materials, then we're going to get differences among the organisms of the same species. And in this context, the offsprings or the daughter cells that were produced are different from their parents. So in the case of the Spirogyra, we have filament A and filament B. And once the cells did release material from the minus to the plus of the filament, we're going to get differences. So we're also going to get recombination. It means we're going to get genes within different parents mixing together. There's also supposed to be hybridization, meaning mixture of two different parents. And rejuvenation in this context simply means that if the genes have lost its vigor, it will be rejuvenated. Then we did mention that this occurs when the environment is not conducive. So it is a way of surviving adverse conditions such as drought. So if the ditch, the pond, or the freshwater environment is drying out, conjugation must occur so that the zygospore will be there for some time. Then if we got water in its environment, then it will begin to germinate. Then the zygote is capable of colonizing new sites because wind current can blow the zygospore away from the dry original environment to a different place. So if we have the zygospore at point A and it was blown away to point B, presumably, let's say 700 meters away, and originally we had a water body 700 meters away without no Spirogyra. Now we're going to get Spirogyra colonizing a new site or locale. And that is another significance since it can now exist in different environments. So that is significance of conjugation. Now we got features of biological importance, which we've mentioned a lot as far as amoeba, paramecium, and euglena is concerned. So structures on the organism playing a very important role, and biologically, the scientists can easily identify. So the first one is the mucilage, the outer covering of the cell wall. And we did indicate that it is protecting the Spirogyra filament from mechanical injury. So in its environment, because they are crowded together, water currents can allow the cells of the filament to slide over each other. And if it is to be cell wall, it will have been dangerous in the context that the cell wall is very hard. Scratching on another cell wall of another cell can create some form of damage. So the mucilage will slide over, and that protects each individual cell from mechanical injury. Then secondly, the spiral chloroplast is for food production. Then the pyronoids are there for food storage. So these are the features of biological importance. And I've told you, anytime feature is mentioned, you got to do with a part, and you need to mention the function. So mucilage protection, chlorophyll food production, pyronoid food storage. Then how the Spirogyra can adjust, can adapt to its environment. One, it got a chlorophyll for food production, because once it can manufacture its own food, it can survive. Then the slimy body or the mucilage is protecting it against injury and at the same time against desiccation, or simply put, drying out. Because if it did dry out, it's going to die anyway. Then the light weight of the Spirogyra filament allows it to float on water instead of being submerged. And that is another way of surviving its habitat. Then the thread-like nature of the filament, not the cell, enables it to entangle to objects within the aquatic environment. So if the Spirogyra is existing in a flowing water body, it will not be carried away by the water current. So its thread-like filamentous structure status prevent it from being washed away or being blown away, because easily Once it is around an object, the other side might be moving away as a result of water current, but actually it can move from its exact location. So these are adaptations to its environment. Any question? Any question? Because if you got no questions, I got a lot of questions for you. Okay. Then let's roll with my question. And I'm hoping we finish with all the questions for everything to be on this video. Now, the first question. Mary, what's the phylum of Spirogyra?
Chlorophyta.
Very good. Now, Mary, give me reasons why you believe Spirogyra is supposed to be part of the Chlorophyta phylum you did mention.
The first one is because the Spirogyra are filamentous. They have no roots, and they are freshwater organisms.
Okay.
And has no cell wall.
Very good. Now, you didn't mention no roots, no cell wall. They are filamentous and they got the spiral chloroplast. So these are reasons why we have organisms placed in that particular phylum. Now, Christian, since your mic is working, state any 3 features of Spirogyra and 1 habitat. And when you check it out, it's 4 marks. It means 3 for the features and a mark for the habitat. So, Christian, answer question 3.
So the habitat, fresh water, and then the features of biological importance. There's chlorophyll for photosynthesis, mucilage for protection of the cell wall, and pyrenoid, food storage. Yes.
For food storage. I like that. I like the answer. Now, then give us any 4 significance of Spirogyra conjugation. Alyssa, just try it. Let's see if your mic will work. If it doesn't, then we'll give the question to someone else.
Zygote is protected. Means of surviving adverse weather conditions or drought.
I like that.
Means about variation.
Very good.
Zygote is used in colonization, new site by the spurs.
I like that. So Callista did give us 4 significance of conjugation. I'm very, very happy I could hear a voice now. For a mark, what is the mode of nutrition in Spirogyra?
It's photosynthetic.
Very good. Continue with question 6. 3 features of adaptation of the Spirogyra.
So chlorophyll for photosynthesis.
2 more.
It's lightweight, so it helps you to float on water.
1 more.
Mucilage for protection.
All right. Now, I'm happy for your question 2, because the 3 answers you did give in an exam, you scored 2 out of the 3. And in WASI, part is a mark, function is a mark. So you mentioned 3 parts, so you scored 3. Then the functions were 3, but you're going to score 2 of the 3 because you use a particular word, this word, which I've indicated that it is not acceptable. This word or its cousin, aid. Because when you mention the part, you say it helps. We don't accept the word help or aid in biology exam at the secondary school level and at the WASI level, which is the end of secondary school level anyway. So never ever use the word help as far as function of a feature is concerned. Christian, is that clear? and the rest of you. This goes to all of us. So don't ever use the word help or aid in doing something as far as function of a feature or a part of an organism is concerned. So Christian, are you okay with that? And this is as a result of your second answer. Christian, are you cool?
Yes, please.
Very good. Now, the reason why we're doing some calculation as far as binary fission and fragmentation is concerned is because of question 7. Now, Mary, read question 7.
A Spirogyra filament is made up of 38 cells. If fragmentation occurs between cells 8 and 9, and 22 and 23, how many?
Okay. Continue.
A. Daughter filaments are formed. B. Cells are present in each daughter filament.
Very good. All of you, you have 1 minute. If you finish, you can just tell me the answer for A and B right now.
Daughter filaments are formed.
3 daughter filaments. Before you continue, how did you get your 3?
I drew the line.
Okay, I like that. Continue.
So for the first one, there are 8 cells. The second one, 14. And the last one is 16. 16 cells.
Okay. So you said for the first one, 8, right? And the second one is? Christian, I'm listening.
First one is 8, second is 14, and the third is 16.
1 5.
1 6.
1 6. So, Calista, 8 plus 14 plus 1 6 is giving me 38. And that is the 38 cells. Now, what Calista did was that he drew a line between cell 8 and 9, giving him automatically 8 cells for the first set. Then between 9 and 22, we are having 4 cells out there. And right after cell 23 to cell 38, we are getting the additional cells of 1 6. And that is how come he had the figures he did mention. Calista, do you understand what Christian did? Mary, do you understand it?
Yes, please.
All right. How are you, Calista? And Ivan, I also want to hear your voice, but your mic has been muted all throughout. Calista, do you get it?
Yes.
Very good. So that is 9 marks. So the first 3 goes for A, and the second 6 goes for B. So that is Spirogyra. There is a question which is not on this device. Okay, let me dictate the question. You guys should write, and let me test your brains on that. I gave it to the Form 1s in their exam. I don't remember exact stuff, but I'm going to try and give you an idea. So let's say a Spirogyra filament is made up of 40 cells. So let me pick a blackboard. So a Spirogyra filament is made up of 40 cells. Out of these 40 cells, 4 did undergo binary fission. So 4 cells out of the 40 did undergo binary fission. Then After the binary fission, I repeat, after the binary fission, fragmentation occurred between cell, let's see, 8 and 9. Then we got another binary fission occurring between cell 20 and 21. Then another binary fission did occur between cell 33 and cell 34. Then finally, we are having another fragmentation. Did I say binary fission? So all that I'm writing is for fragmentation. So the last fragmentation did occur between cell 41 and cell 42. So the question is, how many cells resulted after the binary fission? And secondly, how many daughter filaments have been formed after the fragmentation? Then the third question is, how many cells are in each daughter filament? I repeat the question. So initially we had a Spirogyra cell of 40, filament of 40 cells. And out of the 40 cells, we had 4 cells at random undergoing binary fission. We had 4 cells undergoing binary fission. After the binary fission, fragmentation occurred between cell 8 and 9, 20 and 21, 33 and 34, and 41 and 42. So the question is, how many daughter cells are in each daughter filament, and how many daughter filaments have been formed after the fragmentation? Any one of you, if you finish, let me know your answer.
Hello.
Yeah, I can listen to you.
Yeah, so 5 daughter filaments will form.
Okay, how do you get your 5?
I see that.
I like that. Okay, continue.
The first one has 8 cells.
Okay,
The second has 12. The
how
third
do you get
has,
12?
I counted
Okay, continue.
from the first division to the second. So the
Okay.
third has 13.
Mm-hmm.
The fourth has 8. And then the last one has 3 cells.
Okay, when you added your 8, 12, 13, 8, and 3, what was the total?
44 cells.
Okay, why 44? Because the original filament was having 40 cells.
Yeah, so when the binary fission occurs, there's an additional 4 cells.
Okay, I like that. Mary, do you understand?
Yes, please.
I like that. Ivan, do you understand? If your mic ain't working, type and let me know whether you understand. All right, I have another way of framing the question. Next week test, you guys will experience some of it. Any question? Okay. Since our time is up, I'd like us to end here. And on Friday, we're going to handle our last topic, which you can see currently on your screen, which is the ribosome. So that will be our next topic on Friday. So in the absence of any question, then enjoy the rest of your afternoon. All right, so bye bye.